Hi, everyone. I'm Alexa Fredston. I'm a postdoc at Rutgers University, and today I'm presenting work from my postdoctoral research with Malin Pinsky that's a collaboration with Daniel Afondo and Andrew Allen. Species are on the move. Species in all major taxa and regions have been documented shifting their ranges upward in elevation on land, deeper into the oceans, and generally towards the poles. This global reorganization of biodiversity has been attributed to temperature change due to human-caused greenhouse gas emissions that have altered the climate. This map shows where range shifts in the global oceans have been documented. The shrunken regions are relatively data poor, and the degree to which those range shifts have been consistent with temperature change. The darker blue, the more species are tracking temperature change. But it's one thing to attribute shifts in a species distribution to a driver like temperature, and quite another to predict based on that driver. In ecology, our ultimate goal is often prediction. Sometimes that means predicting the past in order to test whether the models that we fit to data have skill in predicting ecological dynamics. But increasingly, we're trying to forecast the future to provide advice that people might actually act on for everything from tourism development to natural resource management and urban planning. If we could perfectly predict where species will go in the oceans based on temperature, this would be much shorter talk. Unfortunately, we aren't that lucky. These are plots from my previous work on range edge dynamics in marine species in North America. On the x-axis, you can see how the edge shifted over time. Anything to the right of the 0 vertical dash line is a poleward shift, positive, in the direction we'd expect in a warming world. Anything to the left of it is a negative shift towards the equator. And on the y-axis, you can see how much temperature shifted where those range edges were found. If every range edge were perfectly tracking temperature, it would fall on the horizontal 0 line. It would not experience any temperature change, either in the summer on the left panel, in the winter on the right panel, or both. And as you can see, there is a lot of clumping around that 0 line. A lot of them are maintaining the same temperatures, but many of them aren't. This relationship between temperature and edge position might be sufficient for some purposes, depending on exactly what you want to predict. But for a lot of goals in natural resource management and conservation, we need to have really accurate and precise forecasts of species on the move that you might not be able to generate from this type of a correlative relationship between where species have been found in the past and the environment. The way forward that's been proposed in forecasting biodiversity change is process-based modeling, where we explicitly link the outcome or quantity that we're interested in my case, it's species distributions, with a driver like temperature through some mechanistic process. The processes that I'll be talking about in the process-based species distribution model I'm developing are dispersal, demography, physiology, and the environment. We haven't yet incorporated species interactions and evolution, although hopefully you'll see that this is a flexible enough approach that in theory, someone certainly could in the future. These process-based species distribution models are also called dynamic range models. And this is a conceptual slide to walk you through how they work. We start with data on species distributions and abundances, the same way most species distribution models start. But then we write down a process model that describes how we think this population works from a mechanistic standpoint. It might have dispersal or growth or recruitment and reproduction, some of which might be temperature dependent, some of them might be spatially dependent. And once we have that model in hand, we can fit it to these data to estimate the key parameters in the model. Once we have those estimates in hand, we can simulate the future. If we have a forecast for the important environmental drivers in the model or other processes like climate or the presence of another species, we can plug those in along with the process rates we just estimated in this first step and make a forecast that also gives us back what we're interested in, which is future occurrence and abundance. Of course, if this works, it would be really great. First of all, we'd get these interesting parameter estimates that might be informative or useful. Because the model that I'm creating in most dynamic range models are demographic in nature, sort of at the core, it's a population dynamic model. They have the ability to capture transient population dynamics that feed back with distribution, which not all species distribution models can do. For all those reasons, these might be better able to project into no analog climates. Traditional species distribution models often assume that the future habitats occupied by a species will look like the historic ones occupied by the species, or at least their preferences will be the same. And here, because we're using functional relationships between some process rates in the model I'm about to present to you, it's recruitment and temperature, that allows us more ability to forecast into climates the species hasn't encountered before, so long as we understand that functional relationship. For all these reasons, these models could be really useful at near-term forecasting. The model I'm working on is at the 1 to 10 year timescale, which is fairly tailored to what managers often ask us ecologists to project. But there are also a lot of reasons why these models aren't widely used yet. They're enormously data hungry. If you imagine trying to estimate everything that we've described from just species distribution and abundance data in from historical years, you better have a lot of it. Even so, it can be really difficult to estimate parameters. And one reason for that is that you have to choose the appropriate scale for every process in your model. Which ones should vary with time, which ones should vary with space, and at what resolution is actually a very nuanced decision that can really influence how well the models work. And for all these reasons, these models have been primarily demonstrated on simulated data. So this project is one of the first field tests of them on real data, hopefully for eventual operational purposes. I'm not going to walk you through the equations for the model, but instead I'll show you a Bayesian network diagram of how it's set up. This is implemented as a hierarchical Bayesian model in STAN. And this is the process model part, which is what we discussed earlier, how we think the world works in this mechanistic model. The quantity that we want to estimate is in the middle of the slide, the true population size for every age. This is an age structured model, every patch, that's how we did the spatial structure, and every year, which is the time step. And we're saying that that true state is conditional on a number of different processes. One of them is dispersal, which we're estimating. All the dark boxes at the bottom of the slide are quantities that the model estimates. And we also have a couple of parameters in here related to recruitment. Some of them make it a stochastic process because in marine systems where we're field testing this model, recruitment can be very stochastic. And we also made recruitment temperature dependent. So you have terms in here about the optimal temperature, in addition to a process error term. We also passed the model real data on temperature and on mortality estimated in a stock assessment because mortality is really hard for models to estimate. So those are the light gray boxes on the right. Now, if our sampling approach were perfectly unbiased, we could just fit this model to the data, but unfortunately it's not that easy. We're using the NOAA trawl survey data, which I'll mention more in a second. It's not a perfect sample. It has a number of spatial biases, temporal biases, and it preferentially samples bigger fish. So we need another model to capture all of those sampling sources of error so that we can better estimate these true states of the population and these process parameters. That's the other hierarchy of this model, which is the observer model. So here we're saying that the data that we have in hand, the observed number of fish, is conditional on the true state, but also a number of parameters about the sampling process, including one, this length of 50% selectivity that captures how the survey preferentially catches bigger fish. We also have a probability of encountering here because it's a two-stage model that first models whether you encounter a fish at all, and then if you do, how many, and an observation error term as well. So far, this has probably sounded very theoretical, so let's bring it back to where the actual implementation is being field tested, which is the Mid-Atlantic region, although we're using data from the Northeast Fisheries Science Center using this spatial patch structure that I alluded to earlier. You can see the patches kind of drawn in cartoon form on this map, where each patch is 1 degree high in latitude and has within it all of the trawl data from NOAA in their fall survey starting in 1972. This project is actually a partnership with the Mid-Atlantic Fisheries Management Council to hopefully eventually inform their climate-ready fisheries work. And with them, we've identified 4 focal species for which they are really interested in understanding their range dynamics and the possibility of them shifting in the near term. I'm going to talk about summer flounder today because that's the model that is furthest along. But for all of these, we're using data from 1972 to 2006 to fit the model. and then 2007 to 2016 to test the model. Here are some preliminary results and figures. I've put this no tweet symbol on these slides just because these are somewhat hot off the press and I wouldn't want anyone to interpret them as the final forecast. They certainly aren't. These are the actual data. On the y-axis is the patch number, and there's a little map there to help you figure out which one is which. The color is the number of fish total of any age, and brighter colors are more fish. On the x-axis, you have time, and the first year is 1972, so you can, because these are brand new, I didn't change the years to the actual years, but you can add up from there. And there are a couple features in these data that are important for us to capture in our model. One is that in the middle of the time series, you can see that overall abundance is just really low. It's all dark blue, and that's due to overfishing. The second feature is that in the last third of the time series, you see this bright spot appear in the upper right, which is the northern regions where the species predominantly recovered. So in recovering from overfishing, it shifted north very rapidly. So we fit our process model with the observer model hierarchy to these data and then asked, what do you think was the true population size in every patch and year? Here's what our model said, which, as you can see, is not a perfect reconstruction. It does capture that generally they're more abundant in north and south, and it also captures that decline in the middle of the time series, but it's failing to get the recovery at the end, probably because it's mistaking a recruitment pulse for recruitment failure, which is something that's been quite hard for us to tease apart, and I'm happy to talk about more if you have questions. Of course, we're really interested in range shifts, and so we can calculate summary statistics like the centroid position, the weighted range center along latitude. The x-axis here is the patch, essentially. And you can see that, especially in the last third, those red dots, which are the real data, really shifted north. And while our model captures some of the ups and downs before then, it doesn't capture that rapid range shift late. So just for fun for you, I ran a forecast for the final 10 years that are testing data. So here we're not showing the model any of the data. We're just asking it to run forward with the parameters it estimated and the temperatures that we know occurred and tell us where it thinks the population is and at what abundance. And the big plot is our estimate. The little plot off to the left is the true data. And you can see once again, the model's generally capturing that they're more abundant in the north, but totally missing that there's this big population pretty far north that, if anything, only grows more abundant in these final testing 10 years. So we have some more work to do here. One way that I'm hoping will solve the challenge of getting the model to reconstruct the important features of these data is changing where temperature dependence is. In the model formulation I just showed you, we had temperature dependent recruitment, but we might try something like temperature dependent mortality to see if it does a better job of explaining the data. And even if we don't do that, we'll probably revisit the way mortality is being estimated just in case that helps the model along. I didn't mention this too much, but we're also planning to compete these forecasts when they're slightly further along against other modeling methods like correlative species distribution models. So we'll be able to tell which ones perform better at estimating key features of the range, like the range centroid or range edges, every year for the 10 years of our testing period. And finally, of course, this is our first species, and so I have a number of other species to which we'll extend the model. But we're optimistic that in a fairly short time, we'll have both a stronger forecast for summer flounder with a moderate amount of skill, but also hopefully operational forecasts for multiple species that we can eventually use to manage species on the move. Of course, since this is a recorded talk, we can't have a Q&A now, but I certainly hope you follow up with me if you have any questions or would like to discuss any of this research. And in the meantime, thank you so much for your attention and to the Lenfest Ocean Program for funding this research.